Today's lesson is about mitosis, so we're going to be asking ourselves how this process occurs and what the stages are. The learning objectives associated with today's lesson should be quite familiar to you. A lot of it should just be revision from middle school. However, it's important that you are able to use the terminology correctly, and you should also be able to name the stages of the cell cycle in relation to mitosis. These are the key words associated with mitosis in today's lesson. Again, as always, it's important that you're able to use the terminology in the correct way when answering questions. So first of all, we're going to be looking at why mitosis is important. Well, we start off life as a fertilized egg cell, which is a zygote. And within 5 days, so that's less than a week, we've gone from one single cell through to a ball of cells that's starting to become quite specialized. Now this inner cell mass that you see here at day 5, this is what will become us. Now, as early as day 5, we've produced enough cells to make 3 separate potential germ layers. So mitosis is really important for growth, as you can see from these pictures, also for replacing damaged or worn out cells. And it's important to know that this process doesn't just happen once, it is a continuous cycle. However, we do break it up into stages just so that it's easier to understand. So the cell cycle is everything that encompasses the cell division of the cell itself, preparing new organelles, and DNA replication. So the cell cycle maintains growth of cells. So if you think about hair and skin cells, they're continually growing throughout our lives. We are also repairing and regenerating damaged cells. So we know that cell can be damaged through mechanical damage if you have to scrape your hands and so on. And also, it's really important that the cell cycle maintains the chromosome number. This means that every single somatic or body cell must have 46 chromosomes. Now, when we look at the cell cycle, we actually do think of it as a cycle here, and we start with the first phase, which is known as G1. Now, G1, S, and G2 are the 3/4 of the cell cycle that make up interphase. Scientists used to think that interphase was a rest phase, but now we know that a lot of the preparation of producing brand new daughter cells happens here. In the mitotic phase, or M phase, we can see those 4 key stages in nuclear division, but it's actually interphase where all of the other work happens. So if we start looking at the first phase, that's G1, or the growth phase. This is where new organelles are formed, new proteins are formed, and we get the general normal metabolism occurring. This process will end with the start of the S phase, or synthesis phase, where we have DNA replication. So it's really important that you understand what occurs in each of these phases. After the S, or synthesis phase, we have G2, and this is the second growth phase, where now that we have all the organelles that we need and the proteins, and we've got double the quantity of DNA, we start to prepare for the mitotic division. The M phase is where the mitotic division actually occurs, and at the end of this process, we have cytokinesis, which is the division of cytoplasm, and hence we produce 2 daughter cells. Now, normally this process will just continue to go round and round and round. However, there are some points at which the cell cycle is arrested straight after cytokinesis. Now, this is said to be in the G0 phase. Now, these cells that have stopped for whatever period of time, short or long, can then restart and enter the G1 phase of the cell cycle again. So before we look at the chromosomes or before we look at mitosis and how the chromosomes relate, we're going to look at the chromosomes themselves. In this picture, you can see homologous pairs of chromosomes. Now, homologous chromosomes are matching pairs. So when we look at karyograms, that's those picture of chromosomes all ordered from 1 to 22 with the sex chromosomes at the end, we often see two together. And those two together come from the female and the male genetic parents. Normally, chromosomes exist as single chromosomes, as you can see here. But as these chromosomes enter the synthesis phase of the cell cycle, they will undergo DNA replication. And these two homologous chromosomes are now double chromosomes. So I still have two chromosomes here, as you can see above, but these chromosomes here are double chromosomes. And a way of distinguishing them from the original chromosome is that each half of that double chromosome is referred to as a sister chromatid. Now you can't see it in this diagram, but the chromatids are held together by a protein in the middle that would be here called the centromere. So just to recap, DNA is organized into chromosomes, and all sexually reproducing animals will have homologous pairs of chromosomes. For humans, that is 23, whereas in other animals it can be 19 for lions or 39 for African wild dogs, 3 for mosquitoes, and interestingly, some fern species have up to 500 to 600 pairs of chromosomes. So you don't need to learn that information, you just need to know the number of chromosomes for humans, but it does give you an appreciation of the genetic diversity on the planet. Okay, so now we're going to be looking just a little bit more at this idea of chromosomes. When we look at this diagram here, we can see that there are 4 pairs of chromosomes. 2 of them come from the paternal parent, color-coded blue, and 2 of them come from the maternal parent, color-coded red. Now, prior to cell division, in the synthesis phase, chromosomes will undergo replication. So I still have 4 chromosomes, only this time they are double chromosomes. So it's the original chromosome plus a copy. So what we have is 1 double chromosome here, and it's split into 2 sister chromatids. Now this is important in mitosis so that every daughter cell, remembering that we produce 2 daughter cells, each cell will have exactly the same chromosome complement as the original parent cell. Now here in this diagram, I'm just showing you that double chromosome is held by a centromere in the middle, and each double chromosome is made up of 2 sister chromatids. Right, so now we're going to be looking at the process of mitosis, and it occurs in 5 stages. Well, the cell cycle occurs in 5 stages, mitosis is 4. So interphase, remember that's the G1, S and G2 phases of the cycle. This is where DNA replication occurs. When we get into the mitotic or M phase, this is mitosis. And this starts with prophase, and then metaphase, anaphase, and telophase. Now, good mnemonic for mitosis is PMAT, and that always creates a strong visual with me, so I always think about a dog on a little wee mat. So that might help you, might not. Now, it's really important to remember that mitosis is part of the cell cycle, hence it's continuous unless the cycle is in an arrested state at the end of the M phase. However, it will start again at some point. Here we are showing a schematic of the cell cycle. Remember, starting at G1, going into S, G2, and then the mitotic phases where we have prophase, metaphase, anaphase, telophase, and then cytokinesis. Now, you can see here that mitosis is only a really small part of the entire cell cycle, but it does a really important job in ensuring that the resulting daughter chromosomes have the exact same chromosome, so they're replicated exactly, and they have the exact chromosome number as the original parent cell. So now we're going to talk through the process of mitosis and how that works. So we start off with interphase. Now this is obviously just a cartoon, and this is an electron micrograph. Now you can see here that we have this dense material in the middle, and it's indistinguishable. And that is just simply telling us that the chromosome is existing as unwound chromatin, and it's not quite condensed yet. So interphase is where the G1, S and G2 phases of the cell cycle occur, and DNA is replicating at this point. The cell will enter the reproductive cycle with 2 copies of each chromosome. So this, remember, means that those single chromosomes now have doubled. And what that also means is that we will have 22 double homologous chromosomes and 1 double pair of sex chromosomes to make up our 23. The next stage in the cell cycle is prophase, and here we can see that that nuclear material is starting to condense, so that chromatin is starting to supercoil and form visible chromosomes. So these visible chromosomes are now double chromosomes, and what then starts to happen is the nuclear membrane will break down, and centrioles, noted here, will start to migrate to opposite poles of the cell. The next phase in the M phase of the cell cycle is metaphase. Again, you can see in this diagram here, all of our chromosomes have aligned themselves along the middle of the equator. So metaphase for middle. The chromosomes line up along the middle, and what then starts to happen is that spindle fibers start to come out of the centrioles. These spindle fibers will attach to the centromeres, which would be found in the middle of that double chromosome. So metaphase is when the chromosomes line up along the middle of the equator, and centrioles from spindle fibers attach to centrioles. Third phase in the M part of the cell cycle is anaphase. Now I always think about anaphase as being away. So what starts to happen here is these spindle fibers start to contract, and because they are attaching to the centrioles in the middle of these double chromosomes, what they do is they pull those chromosomes apart, and what we have is an identical copy of each chromosome being pulled to opposite poles. And you can see that in this diagram up in the top corner. Final phase of the M part of the cycle is telophase. What starts to happen in telophase is that a nuclear membrane will reform around our 2 clusters of chromosomes, and that nuclear material starts to unwind itself and to relax, so that those tightly wound up chromosomes will then start to relax into chromatin, and we can't see it very clearly anymore. Now, after telophase, the next and final part of this cycle is cytokinesis. So the word cytokinesis, you could break that up into cytoplasm and kinesis, which means to move. And what starts to happen is the cytoplasm will nip in at the top and the bottom here, and eventually it will pull apart. So at the end, we will have 2 separate and identical daughter cells. Now, both daughter cells contain exact copies of the genetic information found in the parent cell, which we know is important, and that concludes the first cycle of mitosis. Now again, this will be repeated continuously, and more cells will be produced in a normal situation. Okay, so that's the end of this lesson on mitosis. Before we finish up, I'd like you to have a look at this diagram and see if you can describe what's happening between stage 1 and 2 in interphase, and describe what's happening between stages 2 and 3. The skill here is to be able to use all the key terminology that we've discussed, incorporating everything you know about the cell cycle and mitosis in your answers. Okay, once you've done that, I'd like you to consolidate your learning by reading pages 249 to 252 of your textbook and completing the notes on pages 13 to 15, including the textbook questions.